Hello students, welcome to SWAM course on microbial technology. Today's topic is cellular component of bacteria. I am Soumya Khare from Chhattisgarh Swami Vivekanand Technical University, Durg. Topic discussed in previous lecture. We studied on bacterial morphology. Describe morphology or shape of bacteria cell is the most distinguished property of a bacteria. Explain morphological trait of a bacteria cell are an important factor in its ability and evolution. Deal with various features like motility, mode of nutrition that are affected by the shape of bacteria. Topic to be covered in this lecture. After completion of this course, the student will able to understand and the student will gain knowledge about the different cell organelles of microorganisms and their detailed function. Student will understand the structure and the purpose of basic component of prokaryotic, especially macromolecules, membranes and organelles. Students will understand how this cellular component used to generate and utilize energy in cells. The content of today's topic is introduction, characteristics of prokaryotic cells, bacteria, structure of bacteria, cell morphology, structure and function of cell wall, gram positive, gram negative cell wall, the bacterial cytoplasmic membrane, nutrient storage structure, capsules and slime layer, flagella, endospore, glycocalyx and summary. Start with introduction. Cell is the basic structure and the functional unit of all known living organism. It is the smallest unit of life that is classified as a living thing and is often called the building block of life. Some organisms such as most bacteria are unicellular, consist of single cell. Other organisms such as humans are multicellular, consist of multiple cells. Humans have an estimated 100 trillion cells. The examples of the cells are unfertilized Orchis egg cells are the largest known cells. The prokaryotic cells such as bacteria and virus and eukaryotic cells are the two basic types of cells, plant cells and animal cells. Bacteria are unicellular, prokaryotic organisms having lacking of chlorophyll. Bacteria can have diameter of 0.2 to 1.5 mm and length of 3 to 5 mm. Characteristics of prokaryotic cells. They lack a nuclear membrane, mitochondria, Golgi body, chloroplast, lysosomes are absent in it. The genetic material is present on the single chromosome. The histone protein is the important constituent of eukaryotic chromosome which are absent in them. The cell wall is made up of carbohydrate and amino acids. The plasma membrane acts as the mitochondrial membrane carrying respiratory enzymes. They divide asexually by binary fission and the sexual mode of reproduction involve recombination. Now we study about the cellular component of bacteria. Some simple prokaryotic cells that may take on a variety of shape are called bacteria. The component of bacterial cells are single circular DNA molecule found in nucleid, cytoplasm, ribosomes, plasmids and a cell membrane. In addition, certain bacteria contain extra features like pile for the attachment and the flagella for the movement. Bacteria are protected and given structural support by their hard cell wall which is composed of a polymer known as peptidoglycan. The size and shape of bacterial cells can differ greatly. Escherichia coli is a popular example. It is a rod shape with dimension of roughly 2 to 3 mu in length and 1 mu in width. Bacteria, however, can take a variety of morphology which such as spiral, filamentous, spirally twisted structures. Structure of bacteria. All cells have 4 main components. DNA, where the residing cells are called the nucleoid region carrying genetic instruction. Surrounding membrane is called the cytoplasmic membrane which limit access to the cell interior. Cytoplasm between the DNA and the membrane where all metabolic reactions occur. Ribosome especially for protein synthesis. Bacteria also often have these features. In this structure, we see that the cell wall which resists the osmotic pressure, the long elongated tube like structure is called flagella, which help to support the movement of bacteria, and the pile, which are the appendages of the cell structure, is used for the attachment and capsule and the protection of biofilm. Here the blue color area is called the nucleoid, where the DNA is reside, and after that the ribosomes in a small molecules are present in the structure, which used for the protein synthesis. Bacterial morphology. Perhaps the most elemental structural property of bacteria is cell morphology, often called the shape. The typical example includes coccus, which is spherical in shape. The examples are Staphylococcus, Staphyloneumoniae, Staphylococcus fruinans. The filamentous structures, which is called the Bacillus anthracis, which is a rod shape, and Haemophilus influenza. And the curve in a spiral shape is the Vibrococci, and this structure is a rod-in like structure. Bacteria display a wide diversity of shape and size called morphology. Bacteria cells are about 1/10 of the size of eukaryotic cells and are typically 0.5 to mu micrometer in length. Most bacterial species are either spherical called cocci, which is single coccus, or rod shape called bacillus, commonly known as bacillus. The elongation is associated with the swimming. Some bacteria are called vibroi, are shaped like a slightly curved rod or comma shape. Other can be spiral shape called spirilla, or tightly called spiroslides. Cell envelope. The cell envelope is all the layer from the cell membrane outward, including the cell wall, the periplasmic space, the outer membrane and the capsule. All free living bacteria have a cell wall. Periplasmic space and the outer membrane are found in gram negative. The capsule is only found in some strain. The bacterial cell wall differ from that of all other organisms by the presence of peptidoglycan. Peptidoglycan is made up of polysaccharides backbone, consists of alternating N-acetyl mimeric acid, also known as NAMA, and N-acetyl glucosamine, also called NAGA, residues in equal amount. Peptidoglycan is responsible for the rigidity of bacterial cell wall and therefore the determination of cell shape. It is relatively porous and not considered to be a permeability barrier for small substrate. Cell membrane. The cell membrane is composed of two layers of phospholipid. The phospholipid have polar head and non-polar tail. Polar head implies that the head are hydrophilic. Hydrophilic means they like to be stay in aqueous environment, facing the outside world and inside of the cell. Non-polar means that the tails are hydrophobic. They want to be away from the water or like the atmosphere in oily environment. The tails are the center of the membrane. In this structure, we can see that the bacterial cytoplasmic membrane is composed of phospholipid bilayer that are all the general function of cell membrane, such as acting as a permeability barrier for most molecule, serving as the location for the transport of molecule into the cells and energy conversion. The head is the hydrophilic and the tail is the hydrophobic. This commonly called phospholipid bilayer. As a phospholipid bilayer, the lipid portion of the outer membrane is impermeable to the charged molecule. However, channels called porine are present in the outer membrane that allow for passive transport of many ions, sugar, amino acids across the outer membrane. These molecules are therefore present in the periplasm, the region between the cytoplasmic and the outer membrane. The periplasm contains the peptidoglycan layer and many protein responsible for substrate binding or hydrolysis and receptions of extracellular signals. Membrane protein. Protein float in the membrane like a ship on the surface of the sea is called fluid mosaic model. The transport protein are very selective. Each types of molecule need their own transportator. Membrane protein are the two type, peripheral membrane protein and integral membrane protein. This is the image of peripheral membrane protein are bound to the surface of the membrane. Some attached to the cell membrane by a fatty acid, covalently attached to one of the protein is the amino acid. Other are attached by stress of hydrophobic amino acid of the protein surface. This image shows the integral membrane protein are embedded in the membrane by one or more stretch of hydrophobic amino acid. A polar is the head and the non-polar is the tail. It composed of the lipid. So, and they are embedded in a multipolar integral membrane protein. Structure of the bacteria cell wall. There are two main types of bacteria cell wall. First, gram positive and second gram negative, which are differentiated by their gram staining characteristics. For both gram positive, gram negative bacteria, particles of approximately 2 and mn can pass through the peptidoglycan. This is the image shows the cell envelope is composed of the plasma membrane and cell wall. As in other organism, the bacteria cell wall provide structural integrity to the cell and determination of the cell shape. While all bacteria cell wall contain peptidoglycan, not all cells wall have some same overall structures. Beta lactam antibiotic such as penicillin inhibit the formation of peptidoglycan cross link in the bacterial cell wall. The enzyme lysozyme found in human tears also digests the cell wall of bacteria and in the body main defense against eyes infection. Gram positive cell wall we discussed in detail. The peptidoglycan is made up of mucopeptide, glycopeptide and murines are the structural element of almost all bacteria cell wall. They constitute almost 95% of the cell wall in some gram positive bacteria. The murine may consist of some as layer of 40 layer and 15 to 80 nm thick, and it's little bit has a 5 to 10% of the cell wall in gram negative bacteria. This is the structure of gram positive bacteria cell wall. Gram positive bacteria made up of peptidoglycan, polysaccharides backbone consists of alternating N-acetyl mimeric acid and N-acetyl glucosamine residue in equal amount. The cell wall of some gram positive bacteria is completely dissolved by lysozyme. The additional component of gram positive cell wall is teichoic acid, a glycopolymer which is embedded within the peptidoglycan layer. This image having the teichoic acid in between the peptidoglycan layer, and this is the outermost layer of cytoplasm. It constitutes the cell membrane, peptidoglycan and lipoteichoic acids. Teichoic acid is believed to be the play several important role of the cell, such as generation of the net negative charge of the cell, which is essential for the development of proton motive force. Teichoic acid contribute to the overall rigidity of cell wall, which is important for the maintenance of cell shape, particularly in rod shaped organism. There are also evidence that teichoic acid participate in cell division by interacting with the peptidoglycan biosynthesis machinery. Lastly, teichoic acid appear to be play a role in resistant to adverse condition such as high temperatures and high salt concentration, as well as beta lactam antibiotic. Teichoic acid can either be covalently linked by peptidoglycan or connected to the cell membrane via lipid anchor, in which case it is referred to as a lipoteichoic acid. Now, we can understand about the gram negative cell wall. Unlike gram positive cell wall, the gram negative cell wall contain a thin peptidoglycan layer adjacent to the cytoplasmic membrane. This is responsible for the cell wall inability to retain the crystal violet stain upon decolorization with ethanol during Gram staining method. This is the image of Gram negative cell wall. We can see here the peptidoglycan is very thin as compared to Gram positive bacteria, and this outer membrane and the inner membrane in between them, this is the periplasmic space which have a thin peptidoglycan layer. In between them lipopolysaccharides, protein and purines are present. Lipopolysaccharides, also known as LPS, the lipopolysaccharides present on the Gram negative bacteria. The periplasmic space in between the cell membrane and the cell wall. The peptidoglycan layer is the outer layer. It is preceded by the prions. The protein channels are and its outer surface is covered with lipopolysaccharides, which often antigenic and toxicity property. As the lipopolysaccharides is highly charged particle, the Gram negative cell wall has an overall negative in charge. The matrix substance in the wall of the Gram positive bacteria may be polysaccharides or teichoic acid. The membrane lipoteichoic acids are anchored in the cytoplasmic membrane, whereas the cell wall of teichoic acids are covalently coupled to the murine. Based on the composition of cell wall and staining, bacteria are classified into Gram positive and Gram negative. The Gram positive versus Gram negatives are defined by the structure of cell wall. The Gram stain bind to the peptidoglycan. Gram positive, many layer of peptidoglycan which is anchored to the cell membrane of teichoic acid. The Gram negative, 1 to 2 layer of phospholipid and peptidoglycan layer is the thin layer. Lipopolysaccharides, also called endotoxin. Gram negative cell wall are thin and unlike Gram positive cell wall, they contain a thin peptidoglycan layer adjacent to the cytoplasmic membrane. The chemical structure of the outer membrane lipopolysaccharides is often unique to specific bacterial subspecies and is responsible for many of the antigenic property of this strain. Lipopolysaccharides, also called endotoxin, are composed of lipopolysaccharides and lipid A, which is responsible for much of the toxicity of Gram negative bacteria. It consists of characteristics lipopolysaccharides embedded in the membrane. This image shows the structure of lipopolysaccharides. It consists of 3 regions: polysaccharides determining O antigen, core polysaccharides, glycoprotein portion or lipid A, and all the 3 factors responsible for endotoxic activity. The lipopolysaccharide endotoxin causes from a septic shock for which there is no direct treatment. Now, we can understand the bacterial cell wall functions. The cell wall provides the following functions of the bacteria. It maintains the shell shape. It is essential for cell growth and reproduction. It prevents the bacterial cell from osmotic lysis. It is providing overall strength to the cell. The cell wall can kept out certain molecules such as toxin, particularly from Gram negative bacteria. The bacterial cell wall can contribute to the pathogenicity or disease, causing ability to the cell for certain bacterial pathogens. The next is the bacterial cytoplasmic membrane. The bacterial cytoplasmic membrane is composed of a phospholipid bilayer and thus has all the general function of the cell membrane, such as acting as a permeability barrier for most molecule and serving as a location for the transport of molecule into the cell. However, channels called porine are present in the outer membrane that allow for the passive transport of many ions, sugar and amino acid across the outer membrane. Mesosome, vesicular invagination of the plasma membrane. It is most prominently present in Gram negative bacteria. The principal site of the mesosome is the respiratory enzyme, and the mesosome is the analog to mitochondrial in eukaryotes. The bacterial cytoplasmic membrane also contain, apart from the mesosome, is the nucleus and plasmid. Nucleus, bacterial nuclei have no nuclear membrane or nucleolus. So, the region where the DNA is present is called nucleoid. Genome consists of a single double strand DNA. It is in a circular form and may be open under certain condition to form a long chain. Another important feature of bacterial cytoplasmic is plasmid. Plasmid is also called extra chromosomal material in bacteria. It is a circular capable of autonomous replication. Plasmid is often called triple C, covalently circular closed molecule. This plasmid is used for the transfer from one bacteria to another. It is a revolutionary in the genetic engineering component or genetic engineering vector. The importance of plasmid: their presence confer to certain special characters. The plasmids contain toxigenicity, antibiotic resistance, ability to use certain unusual component as a nutrients. The plasmid is very important in recombinant DNA technology for the transfer of bacteria and the new gene to the text to the bacteria. Capsules and slime layer. Many bacteria secrete extracellular polymer outside of their cell wall. This polymer are usually composed of polysaccharides and sometimes protein. Capsules are relatively impermeable structure that cannot be stained with dye such as India ink. They are structure that help to protect bacteria from phagocytosis and dissections. Slime layer is involved in the attachment of bacteria to other cells or imminent surface to form biofilm. Slime layer can also be used as a food reserve for the cell. Most bacterial capsules are composed of polysaccharides. The example is Capsula pneumonia. A few capsules are polypeptides, example Bacillus anthracis. The function of capsule: the capsule is an anti-phagocytotic that contribute to the virulence, protect against lysozyme, promote attachment of bacteria to the surface. The most important is the Streptococcus mutans, permit bacteria to adhere to medical implant or catheter. Toxicity to host cell, example Bacterocytes flagellin, provide protection against temporary dying, block the attachment of bacteriophage. Flagella. Flagella are the whip-like structure protruding from the bacterial cell wall and are responsible for bacteria mobility. The arrangement of flagella about the bacterial cell is unique to the species absorbed. The flagella are complex structure that are composed of many different proteins. This flagella contain a protein called flagellin, which make up the whip-like tube, and a protein complex that span the cell wall and the cell membrane to form a motor that causes flagella to rotate. It does not have any ATPase activity. On the basis of the number and the position of flagellum, bacteria are divided into the following form. Atriochus, the bacteria without flagella, is the example is Prosterola. Monotrichos, one flagellum is present at one end, example Vibrio, Pseudomonas, Thiobacillus. Lipotrichos, one or two more flagella present at one end, example is Flagellum spin. Amphitrochus, flagella present at both ends, Nitrosomus, and the Petritrichos, flagella are present in whole body, E. coli, and the example is Clostridium trephini. Each flagellum consists of fibrin and pile. Fibrin are the protein tube that extend out from the outer membrane in many layers or many membrane of the proteobacteria. They are generally short in length and present in high number about the entire bacterial cell surface. Fibrin usually function to facilitate the attachment of a bacterium to the surface and to form a biofilm and the other cells in the form of animal cells during the pathogenesis. Pile are similar in the structure of fibrin, but are much longer and present on the bacterial cell in low number. Pile are involved in the process of bacterial conjugation, and the non-sex pile is aid to bacteria in gripping surface. The pile is mostly used in the conjugation genetic recombination of bacteria. Nutrient storage structure. Most bacteria do not live in environment that contain large amount of nutrients in all time. To accommodate this nutrients level of nutrients, bacteria contain several different methods of nutrient storage in time of plenty for the use in the time of want. For example, many bacteria store excess of carbon in the form of polyhydroxyalkanate or glycogen. Some microbes store soluble nutrients such as nutrients in vacuole. Another form of nutrient storage in bacteria is the inclusion, are considered to be the non-living component of the cell that do not possess metabolic activity and are not bounded by the membrane. The most common inclusions are glycogen, lipid droplet, crystal, pigment, protein vacuole and cytoplasmic inclusions of complex inorganic pyrophosphate. These granules are called metachromatic granules due to their displaying the metachromatic effect. They appear red or blue when stained with a blue dye or methylene blue. Gas vacuole. Gas vacuoles are membrane bounded spindle shaped vesicle found in some placentonic bacteria and cyanobacteria. They are made up of shell of protein that has a highly hydrophobic inner surface, making it impermeable to water, but permeable to most of the gases. The diameter of gas vesicles will also help to determine which species survive in different bodies of water. Endospore. The highly resistant stage of bacteria is the endospore. Spore germinate to form a single vegetative cell. It is formed inside the cells, especially in Bacillus clostridium. Spores contain the nuclear body surrounded by a spore wall. Outside this is a thick spore cortex. It is covered in turn by a tough spore coat. Exosporium in addition outer covering which exhibit rigid and groove. Many bacteria secrete extracellular polymer outside of their cell wall called glycocalyx. These polymers are highly composed of polysaccharides and sometimes protein. Capsules are relatively impermeable structure that cannot be stained with dye such as India ink. These structures that help to protect bacteria from phagocytosis and dissection. Slime layer is involved in the attachment of bacteria to other cells or imminent surface to form biofilm. Slime layer can also be used as a food reserve of the bacterial cell. Summary: bacteria are microscopic single cell prokaryotic organism. Most of the bacteria contain cell wall as an outer covering of the cell membrane. The cell wall provide the following benefit to the bacteria. It maintains the cell shape essential for the bacterial cell growth, movement and reproduction. The cell wall is also prevent from the osmotic lysis. The topic to be covered in next lecture is the sporulation on its mechanics. Sporulation refer to the formation of a spore from vegetative cell during the unfavorable environmental condition. It occurs in different organisms like bacteria and fungi. Spores are capable of preserving the bacterial genetic material when the condition become harsh and difficult for the normal form of bacteria and fungi. These are the references of this topic: Cooper, David H. and Medin for the Brock Biology of Microorganism.